Yasinskaya, Medical Officer in the Division of Anti-Infective Products at CDER, same division Director of. I will be presenting you safety assessments of clinical trials and clinical trials. This is outline of my talk. First, I touch on the sources of safety information is available to you, and then with that information, we'll discuss what questions we need to ask when considering a patient to be enrolled in the trial. Then I will go over safety monitoring and ascertainment of adverse events, as well as adverse events coding, and then I will talk about safety reporting. And I will conclude my talk with a brief mentioning of the market safety. So, product safety as it goes through the development and after approval is evolving process. So basically, it can span from no clinical data, just in vitro and animal studies, to and it continues to past approval collection of safety data when it's the product is used in millions of people out there on the market. The available data depends on the stage of product development, and safety information for the approved products can be found in the package insert, while for the products under investigation, the investigator's brochure. And in both of these sources, the information, you will see the information in vitro safety, as well as non-clinical safety that includes mechanism of action and animal studies. There, depending on the stage of product development, you may find also the data from first studies in humans, and generally these are phase 1 pharmacokinetic studies in healthy human volunteers. And then in addition, there are going to be multiple clinical studies as the development of the product continues. Safety from randomized controlled clinical trials will be available. So the most pertinent data for you when you're conducting a study would be the data from the clinical trials for these similar indications. The baseline characteristics of the patient are going to be very similar to what the patients you're going to be looking at to enroll. However, you need to consider other sources of safety data, such as non-clinical data, the mechanism of action, the chemical structure of the product, as well as and other non-clinical studies, such as in vitro toxicity studies and studies in animals. Clinical pharmacology studies, clinical trial safety data for other indications are very helpful, and the product is currently approved and marketed. Post-marketing experience with the product will be available as well. The other sources of safety information are publications and the safety profile of drugs in the other class come in handy as well. So with regards to the chemical structure or product class, class toxicities such as, chemical structure, if you have an antimetabolite product as a chemical structure or mechanism of action, you would know that most likely the effect would be on bone marrow. If the drug class toxicities, the fluoroquinolones share a lot of safety profile between the products in the class, and for example, peripheral neuropathy is a common adverse event that is common between the fluoroquinolones in the class. In vitro toxicity evaluations generally tell you about carcinogenic to teratogenetic potential of the product, as well as its effect on cardiac regularization. Those toxicities could be further evaluated in studies in animals, and in addition to carcinogenicity and teratogenicity, organ-specific toxicities at first will be manifested in the studies in animals. With regards to the phase 1 pharmacokinetic trials, we learned about the product's absorption, metabolism, and the pharmacokinetic profile. This is helpful in designing the appropriate protocols and collecting safety assessments. These studies are conducted for majority of products in healthy subjects, but in oncology indications, the disease versus exposed to the products. In patients, so where there's an indication where there's a disease present, will help to identify potential drug-disease interactions and especially populations that might metabolize and have a different pharmacokinetic profile from healthy volunteers. That's the pediatric subpopulation, elderly, people with hepatic and renal impairment, and pregnant and nursing mothers. We need to consider drug safety profile in dose escalation trials because here it's the most pure safety assessments are being conducted. Generally, the comparator in these trials is placebo, and in dose escalation you have small doses and you have higher doses. You can see if there's some dependence in adverse events. These studies generally will either support finding safety signals identified in clinical trials, and but they also identify the safety signals that are specific to humans only. So after you consider the breadth of information available to you in the investigator brochure, you need to decide if the patient that you're currently evaluating is fit for the protocol. You obviously need to apply inclusion criteria, and evaluation of medical history and screening laboratory values will help to identify the potential compromised organs that the patient might have, and with what you know about the product, where this toxicity might be exacerbated. Concomitant medications, the knowledge of these are very important as they can potentially share the metabolic pathway with the product under investigations, and potentially can result in higher concentration or lower concentration, and therefore exacerbating potentially the toxic profile of the drug. You also need to have to take into consideration the dose, regimen, and duration of dosing, as well as the duration of follow-up. While you're examining the pharmacokinetic parameters of the product, these have particular implications of how you monitor the subject. You generally will see acute toxicity around the Cmax, so you need also know when the Cmax is observed. And also with the dose escalation, how linear the exposure with the product is, so you know if there's any potential for drug accumulation. You need to know the half-life of the drug to know whether the duration of follow-up is sufficient. So how do we monitor safety in the clinical trials? We collect information about adverse events. And adverse events, there's a following slide where I discussed the regulatory definition is an untoward medical occurrence with when the patient, the subject, is exposed to a drug. There's some association, but there's not necessarily a causality. So there's myriads of adverse events, and they could be very common in general population. As a baseline, headaches and vomiting, diarrhea. It could be extremely rare events that are uncommon in general populations, such as anaphylaxis. We'll go over what the definition of serious adverse events is very important because it's a regulatory definition, and what is an adverse reactions. And the adverse events of special interest, those adverse events that have been identified in the early product development and need to be further characterized in the this particular clinical trials. Laboratory abnormalities could also be adverse events. The adverse events could be ascertained either in passive or an active way. And so they either can be spontaneously reported or observed by the investigator or study personnel. As well, they could be reported as a result of the probe, so active surveillance of the checklist, of questionnaire, or both. So additional safety assessments and monitoring that employed in clinical trials is assessment of vital signs, evaluation of laboratory parameters, as well as special safety assessments as dictated by the previous examination of safety and safety signals. So this is the regulatory definition for adverse event or experience. It's an untoward medical occurrence associated with the use of a drug in humans, whether or not considered to be drug related. So they don't necessarily the drug had caused the event, but it had been observed during exposure to the drug. And it could be a sign, symptom, or disease, abnormal laboratory values, vital signs, as well as findings on the imaging and other investigations, or worsening or constellations of the above. So adverse events, when you're reporting them, and the case report forms, will be graded from mild to severe on a scale. And they generally, there's scales, rating scales, there are a number of them, and they're very specific to the study population, the phase of the product development, as well as the product, depending on the product being evaluated, whether it's a small molecule or it's a biologic, whether it's a device or vaccine. And this is the list of the grading scales that's available to you. Here's an example of an NCI grading scale, common terminology criteria for adverse events, used in development of oncology protocols. And generally, it's not appropriate for the healthy subject, as the threshold for labeling something mild versus moderate versus severe is clearly different between the healthy subjects and patients with oncology indications. Here you can find the adverse event severity grading scale that is used in healthy human volunteers in vaccine trials. And although the grading mild, moderate, severe, potentially life-threatening is sort of similar between the two in labeling the certain grade of the adverse event, but again, the threshold, what we will be able to tolerate in healthy human volunteers versus what is going to be appropriate for oncology patients would be very much different. Serious adverse event, you'll need to know the definition. It's a regulatory term because U.S. clinical investigators would be reporting all serious adverse events, regardless of causality, to the sponsor of the protocol. It's any adverse event that results in your opinion, in death or is life-threatening, results in hospitalization or prolongation of existing hospitalization. Is persistent. It can result in persistent or significant incapacity in conducting activities of daily life, such as disability, or in congenital anomaly or birth defect. Here's the examples of uncommon serious adverse events, and because these events are very uncommon in general population, occurrence of even a single one of them in a clinical trial can raise a suspicion of it being drug-related. How do we evaluate serious adverse events? Because you guys are going to be reporting serious adverse events to the sponsor of the protocol, you will be required to also assess a causality, whether this adverse event that you're currently observing and reporting on is related to the study drug exposure. So you first need to assess whether the event that you're currently observing whether it's common in occurrence in the population on the study. Is the population elderly and myocardial infarction in patients with underlying coronary disease is pretty frequent, or you see cases of myocardial infarction in middle-aged population. So the baseline frequency of adverse events in population will give you the clue, whether it's rare or not so rare. Then you need to assess whether the event is treatment emergent. So by treatment emergent, we mean that it occurred during the exposure to the drug. So, and it could span the product administration. So while the patient was taking the drug, but then also it will be treatment emergent if the administration of the product had already stopped. But you know that the half-life of the product is pretty long. So therefore, in the duration of follow-up, the drug is still in the system, and therefore the event is still considered to be treatment emergent. Whether the adverse event diminished or completely resolved upon the discontinuation of the drug, or whether it recurred on rechallenge when the drug was reintroduced. Were there any concomitant medication that have a safety profile that can potentially could have caused this particular adverse event? And then, what do you have to support the adverse events and things like characteristics of this particular patients, whether they're pertinent laboratory tests or other examinations available and are being done to evaluate how these events have evolved over time, and will help you to identify whether the event has been related to the product. Also, whether there's an obvious alternative cause, and if this serious adverse event is a study endpoint, that is very important because even though you're going to be reporting it to the sponsor will not be reporting it to the FDA as an expedited reporting the adverse event that is a study endpoint. For example, mortality. If somebody died on the study, if this is a study endpoint, it will not necessarily be considered to be a reportable adverse event, unless the mortality was due to a rare event, serious adverse event that among those that have been listed on the previous slide, for example, a death due to anaphylaxis. So your responsibility per our regulation is to report all serious adverse events with assessment of causality to the sponsor. You also will need to report adverse events or laboratory abnormalities that have been identified in the protocol as critical to safety evaluations. And study endpoints, as serious adverse events will be reported that there's evidence to suggest a causal relationship to the product. So please, because you're going to be reporting this to the sponsor, and then the sponsor in turn will be reporting these to us if there's a strong possibility that the event had been caused by the drug. All the pertinent information regarding this event would be very helpful, and that includes any investigation done inside and outside the protocol, whether it's laboratory imaging studies or whatnot, and consults. So when you are recording and reporting your adverse events, you using a certain terminology, but then this your native term will be converted to a standardized term for this, to allow the sponsor and then FDA reviewers to assess safety of the product in the aggregate, to allow the standardized analysis, sorting, and grouping of like events. Preferred term is used to calculate incidence of adverse events. It's a unit, the lowest unit that we are considering for analysis of safety. And the dictionary that we are currently using to code adverse events is MEDRA, Medical Dictionary for Regulatory Activities. On this slide, we have a schematic of MEDRA structure, and as you see, adverse event reported verbatim by you will be basically equivalent of the lowest level term, which then will be converted to appropriate preferred term. That's a very specific, very specific unit, a medical concept, while the highest level of terminology and least specific would be system organ class, for example, infectious infestations or cardiac or urinary and so on. So because there is a certain amount of knowledge about the event, and obviously the knowledge of coding processes are required in order to convert your verbatim term to a preferred term for the analysis, certain problems can arise where the same adverse event could be split between two different preferred terms, for example, hypertension and high blood pressure, even though for clinician it's the same, it could be coded in two different ways in MedDRA. Or there could be a lumping of different terms for the same preferred term of edema, like leg edema and facial edema, even though they have a different pathogenesis, they could be lumped into a single preferred term. That obviously will pose a problem in the analysis of the adverse events later on. So we urge you, when you're reporting serious adverse events, to use, to be very consistent what you're reporting, use the most scientific terminology when reporting them, so it creates less problems in the coding. The next definition I would like to go over is unexpected adverse events. And although the expected reporting is not your responsibility per se, it's responsibility of the sponsor, you might want to be aware of when the sponsor will report certain events to the FDA, and because based on that, the investigator's assurance, the knowledge about the safety profile of the product will be based on. So unexpected adverse event is the adverse event that is not listed in investigator's brochure, or if the investigator's brochure is not available, so basically it's a package insert, for example. It's not listed in the specificity of severity observed. For example, you will have a term of hepatitis listed in the brochure, but what you're observing currently in the patient is hepatic necrosis. So even though it's the same organ, if the abnormalities are sort of similar, but it's a different level of severity, this would be considered an unexpected adverse event. In addition, the adverse event could be considered unexpected when it's mentioned in the investigative brochure is anticipated because of pharmacokinetic properties of the product or chemical structure, or it had been observed with the product in the same class, but had not yet been observed with a particular product that you're currently investigating. Additional term that we need to go over is suspected adverse reaction. A suspected adverse reaction is an adverse event for which there's a reasonable possibility that the drug caused the adverse events. And these could be a single occurrence of an uncommon event known to be strongly associated with drug exposure. You can refer back to the list that I have provided you of rare uncommon adverse events, serious adverse events, like Stevens-Johnson, for example, or anaphylaxis. Or it could be more than one occurrence of event that is not commonly associated with drug exposure, but otherwise is uncommon in the population under study. Again, myocardial infarction in elderly would be common, with coronary disease, with underlying coronary disease, and healthy middle-aged patient will unlikely have this event. Or on aggregate analysis, and this is available obviously to the sponsor, that specific events observed in the clinical trials are occurring more on the study drug as compared to the concurrent control or historical control. This diagram shows you the continuum of the adverse events, and the closer you are to the center, the stronger is the suspicion that the adverse event had been caused by the drug. And so the greatest degree of certainty would be an adverse reaction, where all the factors indicate that the drug caused the adverse event. So expedited safety reporting to the FDA is done by the sponsor, and they collect all of your reported serious adverse events and look through them. They do an independent assessment of whether they consider the adverse event serious, whether there's a reasonable possibility of the product causing the adverse event, and they will identify whether the event is unexpected based on the breadth of knowledge that they have about the product safety profile to date. And they have it from different sources. Obviously, you only have investigators for sure that might be updated, or that is most up to date at that particular time, but they have different streams of safety data that's coming to them from different studies, whether it's clinical, non-clinical publications, and so on. Fatal or life-threatening, serious, unexpected, suspected adverse reactions should be reported to the FDA no later than 7 days upon the knowledge of the event occurring. Other serious, suspected, unexpected adverse reactions should be reported to the FDA no later than 15 days. In addition, the safety findings from other clinical and non-clinical studies need to be reported expeditiously to the FDA if they contain important safety data or signals that will change the product safety profile, and therefore might possibly change the risk-benefit assessment of conducting a certain protocol. They will also need to report the increased rate of occurrence of serious suspected adverse reactions, and this needs to be reported to the FDA within 50 days upon becoming aware of the finding. So the U.S. individual investigators sometimes, you generally will not have sufficient amount of data and knowledge available to you about the product when it applies to common adverse events like nausea, vomiting, diarrhea, headache, myocardial infarction, yet again, in elderly. Such events will require aggregate analysis using population approach, and that is available to the sponsor. So once there's a rate difference of these adverse events been found not to favor the comparator, so the drug on the study will have a higher rate of these events, it will have to be reported to the FDA. So that basically concludes evaluation of safety during when the product is still in development. However, the product doesn't stop development even after approval. And with approval, generally there are additional studies that are being conducted. We draw safety information from the non-clinical studies, additional clinical trials for the same indication, maybe for a different patient population, for different indications, for observational studies, and we have registries depending on how the product performed prior to approval to draw a lot more safety information when drug finally reaches the market. Besides these structured safety collections, there's also FDA adverse event reporting system repository that is collecting safety information about serious adverse events regardless of causality through the MedWatch. We have a vaccine adverse event reporting system, and Sentinel Initiative is currently functioning. It's an active surveillance program to query diverse automatic healthcare data. That's another stream of safety information we have for the product that are currently on the market. I'll touch briefly about the MedWatch. It's an FDA reporting system for adverse events, has been founded in 1993. It's passive, so unless somebody, such as healthcare professionals, consumers, or the patients report an adverse events, we will not know what is the safety profile of the product that is currently being used by millions of patients. To report an adverse event, serious adverse event via MidWatch is fairly easy. It's a one-page form, can be done online by fax or mail or by phone. And if you subscribed, it also provides you with potential safety signal alerts from the FDA. So the the knowledge of product safety doesn't end with approval. It spans the whole life of the drug from the beginning to the end. And you as investigators provide us with the clinical and laboratory and additional test data if available.
And for us to decide whether the adverse event had been associated with the product use is actually an adverse reaction. And please continue reporting serious adverse events to us via MidWatch once the product hits the market. These are the references that are available to you, the regulations and the safety reporting rule with the guidance, FDA guidance on implementing these reporting standards, as well as the toxicity grading table from the FDA and MidWatch.